All right, class, today we're going to start Unit 4 with Lecture 1, where we're going to be discussing protein and amino acid structure. Before we get into the nitty-gritty details of protein and amino acid structure, just going to define what proteins are. So proteins are long-chain polymer compounds that are composed of amino acids. And amino acids are molecules that are composed of carbon, hydrogen, oxygen, nitrogen, and sulfur. Now, not all of the amino acids have sulfur, so the only ones that have sulfur are methionine and cysteine. So you'll need to be able to tell me methionine, cysteine have sulfur, but all other amino acids have carbon, hydrogen, oxygen, and nitrogen. Methionine and cysteine have sulfur. All animal tissue, or most animal tissue, is proteinaceous in nature. It's needed for metabolic reactions. All of our enzymes are made up of proteins, and they are also, the biological properties of the proteins are determined by the amino acids that make up the protein and how they are sequenced together. So our amino acids are what is making up our protein. So our protein is made up of amino acids. And interestingly, there are over 300 amino acids utilized in the plant world. However, animal protein is mainly made up of 20. So we have 20 amino acids that make up animal protein. And the vast majority of those amino acids have additional metabolic functions, but they make up protein and they have other metabolic functions in addition to their protein structure. There are some non-protein amino acids that are important for metabolism in animals, but those non-protein amino acids are not part of the protein or peptide chains. Some examples include ornithine and citrulline, and ornithine and citrulline are major components of the urea cycle, which we'll talk about in a little bit. We also have our amino acids. They are the building blocks of all of our structural and constituent proteins, so the building blocks of our muscles, our cartilage, bones, organs, skin, and hair. All is the basis of amino acids are the basis of those. In addition to amino acids being important for protein structure, they also are very critical for making up regulatory proteins. So many of these regulatory proteins include enzymes. So our enzymes are proteins that act as catalysts for biochemical reactions. So these are reactions that would happen naturally, but we need to encourage those two molecules to come together to have that reaction. So that's what we use enzymes for. In addition, we have hormones. So these hormones are actually peptide hormones or protein hormones that are made up of amino acids. So these hormones are messengers of metabolic signals within the body. They utilize the blood and other body tissues or body fluids to carry out their physiological response. So typically when we refer to hormones, everybody thinks of steroid hormones, but these hormones are different. They're peptide hormones, and they are utilizing secondary messengers. So for instance, insulin is going to bind the insulin receptor, and that's going to send a signal. So insulin is not directly affecting the upregulation of genes or protein expression. So these hormones are working through second messengers. We also have amino acids make up neurotransmitters, and these are chemicals involved in exciting or inhibiting electrical impulses between nerves. And again, there is physiological response within the target organs and tissues. So our amino acids have a general structure. So they all have this backbone structure, which is made up of an amide group. So this amide group is composed of an amino group, an acid group, and a carbon. So our amino group is our nitrogen and hydrogen. Then we have our acid group, our COOH, and then our central carbon. And then you can see they all have a hydrogen conjugated to the central carbon, and we have this R group. And this is R group is what's giving the individual biochemical characteristics. So if we say an amino acid is neutral, it's because the R group is neutral. We still have the charges between the amino and the carboxyl groups are always interacting, and they're always ionized some way. So our amino group may be charged and have a, sorry, this should be a 3 here. I thought I fixed that. So our amino group can have 3 hydrogens on it, and our carboxyl group can have a charged oxygen. Sometimes we can have an H conjugated there, and that really depends on the pH of the solution and what the amino acids are in. And because of this basic structure of the amino acids, we can have different isomers. So similar to how we had the carbohydrates, glucose had a D or an L configuration, amino acids have the same thing. So you could see here our L amino acid is basically a mirror image of our D amino acid. So we can have a D or an L isomer, all with the exception of glycine. And glycine is the exception because the R group is a hydrogen. So they are not going to have this mirror image. So this isomer is all depending on the configuration around the asymmetric carbon. So our asymmetric carbon's in the center here. And in contrast to our carbohydrates, where the D isomers were natural, here L isomers are natural in our amino acids. So these are the only ones that are used in proteins. So you'll never have a D lysine put in used for protein synthesis. In fact, there's only one amino acid that can utilize the D isomer, and that is D-methionine. So this D-methionine first, before it can be incorporated into protein, has to be converted to the L isomer. But we're able to convert D-methionine to L-methionine. So if you ever happen to be mixing feed for animals, you'll notice that the bag for methionine typically says DL-methionine, where your lysine and threonine is going to always say L-lysine or L-threonine, because you cannot utilize D-lysine and D-threonine. So in addition to the isomers that amino acids can form, they also have a Zwitter ion structure. So a Zwitter ion is where depicted here on the right hand side, where we have the amino side is charged. We have the 3 hydrogens, so that has a plus, and our carboxyl terminal is also charged, where our oxygen is not bound to a hydrogen, so it is negatively charged. So that ends up resulting in an internal salt formation between the charges. And because of this, amino acids are considered to be amphoteric substances, meaning that they can react both as a base and an acid, and that all depends on the pH in which they are occurring. And that's all based off of the charge off of the amino group or the carboxy group, not off of the R group. And these charged moieties, they are always interacting with each other. You can see on the left the difference between the Zwitter ion configuration and the non-Zwitter ion. So our non-Zwitter ion is where neither end is charged. So very rarely are you going to be having a non-Zwitter ion with amino acids occur. Typically, we're going to be, depending on the pH, have some either charge on the amino group or a charge on the carboxyl group. So here's a list of the 20 amino acids that we utilize in animal protein. And you can see here, we're going to talk about this a little later, the different categories that these amino acids can be classified in. And it's all based off of the structure of the R group. So we have our nonpolar, our polar, and then we have our acids and our bases. So all of these structures, these biochemical characteristics are all due to the R group, and they all have the same backbone. That amide backbone is the same for all of them. So as I just mentioned, we have the different categories of amino acids. We have our nonpolar, our polar that are not charged. So these are also referred to as neutral. And then we have our polar with charged side chains. And we have two subcategories. We have our acids and our bases. So here we have all the amino acids that are our non-polar side chains. So you don't have to know the structures, but I do want you to be able to tell me which amino acids are non-polar. So be able to tell me that glycine, leucine, tryptophan, alanine, isoleucine, phenylalanine, valine, methionine, and proline are all non-polar side chains. So these are generally non-reactive. In addition, because of the non-polar aspect of their side chains, they are very hydrophobic. So these amino acids are going to be more likely to be on the inside of the protein. In addition, these will be the ones that are going to be on the outside and the cell membrane. Because we want the amino acids that are going to be able to interact with the fatty acids, which is not very conducive to water. So these ones are going to be more apparent in those transmembrane receptors. So the hydrophobic amino acids will be making those up. So our next category is our polar non-charged, and once again, be able to tell me these amino acids that are polar non-charged. And these include serine, glutamine, threonine, tyrosine, asparagine, and cysteine. And so these are polar. You can see that we have, for instance, in our serine, we have our hydroxyl group. We have our amino group on the end of our glutamine. We have another hydroxyl group for threonine, hydroxyl group for our tyrosine, amino group for our asparagine, and our sulfur for our cysteine. So they are polar. Unfortunately, they cannot be ionized, and don't carry an overall charge. But because they are polar, they are still hydrophilic. They like to be in water. Our last category is the polar charged. And like I mentioned before, this category has two subcategories. We have our acids and we have our bases. So here we only have one of the acids, glutamic acid. But we also have aspartic acid. Sometimes they're referred to as glutamate or aspartate, depending on if they're charged or not. So here it's showing in the charged form, so it's referred to as glutamic acid. You put a hydrogen on there and it's glutamate. And then our bases include our lysine. So we have the amino acids also have different categories that we split them up into, and based off of if our bodies can make them or not. If our bodies are unable to synthesize those amino acids, those are referred to as essential, meaning that they must be consumed in the diet. So we cannot synthesize them at all. Our non-essential are ones that we can synthesize if we have the precursors for them. So here's a list of our essential amino acids, which include arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So the asterisks at the arginine, methionine, and phenylalanine, that all is depending on the species. Some of those are conditionally essential, and some, and meaning that the animals can synthesize some of it. If they're unable to synthesize adequate amounts, then it will be essential. Now, ruminants do not have an essential amino acid requirement. That's because they are utilizing the microbes to make the amino acids. So basically, because we do not control necessarily what the microbes are making, so therefore we can't say that they have an amino acid requirement. So if you look in the NRC for beef or dairy, sheep, goats, that's not going to tell you specifically that they have 1.25% lysine requirement. They're gonna have a protein or a nitrogen requirement. So what's a way you can remember these essential amino acids or indispensable amino acids? And the way I've learned and the way I'm teaching you and most everybody that I know has learned is using Private Tim Hall. So that stands for phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, lysine. So all of these ones are the essential amino acids for our non-ruminant animals. And the biggest thing is where people get confused, they may put in tyrosine for one of the T's, but that is actually a conditionally essential or semi-essential amino acid because it can be synthesized from phenylalanine. So threonine and tryptophan, not tyrosine. So when we are utilizing our amino acids for energy, we have to account for the ammonia or the amino group that needs to be removed before we can have it synthesized into glucose. So with that, you can see here within our mitochondria, we have our ammonia bind with bicarbonate, and this requires energy, 2 ATP, and then we get the formation of carbonyl phosphate. This then combines with ornithine, which is one of those non-protein amino acids. So our ornithine is then converted to L-citrulline, another one of those amino acids, with the carbonyl phosphate, and then our citrulline is able to leave the mitochondria, and eventually, with a little bit more energy, we have the formation of our arginine here. So our arginine is then, we take off the urea, we form urea off of the R group, and then we resynthesize ornithine. So this is the way that most mammals are detoxifying the ammonia. So urea is less toxic than ammonia. Now with birds and reptiles, they do not have the urea cycle, or they have very, very small amounts. So therefore this is how you're synthesizing amino acid or arginine. So that's why birds and reptiles have a higher requirement for arginine, or it is essential because they're unable to synthesize it in the urea cycle. If we have a shortage of any of these amino acids, you're going to end up with ammonia toxicity. So let's say we have a situation where there's inadequate arginine in the diet. So we're not going to be able to, we need that arginine for protein instead of the urea cycle. So we're not able to synthesize adequate amounts of it. So we keep on pulling arginine off. Eventually you're going to have this flow back, and you're going to have an abundance of the ammonia here. So then you're going to end up having ammonia toxicity. And we'll talk about the negative ramifications of the ammonia toxicity in the next lecture. So not only is arginine essential for avians and reptiles because of the lack of the urea cycle, it is also essential for the cat. So the cats lack enzymes needed to synthesize ornithine, and that ornithine, if you remember, is used to regenerate the arginine and the urea cycle. So if you feed cats a diet without any arginine, within a few hours, you're going to see signs of ammonia toxicity. So they get hyperammonia. They'll start getting frothing at the mouth and have many of the other symptoms of ammonia toxicity. And in other species, they are able to use glutamic acid and proline to synthesize ornithine, but the cat is not able to. And it makes sense. The cat evolved. It's an obligate carnivore. I should say that this is not just cats. Any obligate carnivore is going to have this similar situation. But we've studied cats well enough. So all of our obligate carnivores are unable to synthesize ornithine for this urea cycle, and it's because they have adequate amounts in their natural diet. Now, dogs, even though they are not obligate carnivores, if you feed them a diet without arginine, you'll still see some signs of ammonia toxicity. It is not as severe as what you observe with the cats. So all the more reason to stay away from the crazy diets that people come up with for dogs. In addition, since cats have a higher arginine requirement, this is another reason why you should not feed cats dog food, because it's not meeting the requirements for the cat. Since the cat is an obligate carnivore, it cannot synthesize arginine. You need to make sure that cats are not eating dog food. Now, dogs can eat cat food, that's fine, but not vice versa. So I mentioned earlier we have these conditionally essential amino acids. So these are amino acids that behave as essential if the rate of synthesis is less than the rate of degradation. And what is conditionally essential varies with species. Typically, you can have a conditionally essential amino acid become essential if you have a shortage of a non-essential amino acid precursor, or sometimes they're even essential amino acid precursors. For instance, you can utilize phenylalanine to make tyrosine. So if you don't have adequate phenylalanine, or you have only enough phenylalanine to cover its requirements, you're not going to be able to cover the shortage of tyrosine. But the vast majority of the conditionally essential amino acids are coming from non-essential amino acids. Usually you're going to see these deficiencies of these conditional essential amino acids when you have a major physiological change in the animal. Most notably, this is going to be related to reproduction. So an animal becomes pregnant. When it becomes pregnant, when it starts making milk, when it starts laying eggs, that's when you're going to start seeing this change, because there's a added requirement. So reproduction is one of the main triggers. In addition, any trauma that occurs. So, specifically, if we're referring to surgery, so surgeries are going to result in major physiological change, and therefore we need to pull our resources, and that's going to make some of these non essential amino acids become essential. One of the ones that becomes essential during trauma is our glutamine. So, glutamine is found in large quantities in muscle, and it is mobilized pretty greatly after surgery, especially if you have intestinal surgery, and that's because it is the favorite energy source for enterocytes. So, therefore, if you know anybody that's ever had intestinal surgery, Crohn's disease has parts of their intestine removed, bariatric surgery where they have parts of the stomach removed. Typically during the recovery time, it's essential. So we put glutamine in the IV for these people. In addition, if you know anybody that happens to have total parenteral nutrition where they're being fed through their IV, we also provide glutamine for them. So any of these traumatic instances, glutamine becomes essential. Now, for our avian species, we have some conditionally essential amino acids, and that includes proline, glycine, and histidine. So this is mainly because of the lack of the urea cycle. So they are forming uric acid instead of urea. So a lot of these are going to components of these different amino acids are being utilized to form uric acid. So these ones be able to tell me that proline, glycine, and histidine are conditionally essential for birds. Another conditionally essential amino acid is taurine. And you may be intrigued because this was not on the list of our amino acids. It's not one of the 20. So, taurine is one of the non protein amino acids. It is actually synthesized from cysteine and is conditionally essential in our obligate carnivores. So, taurine is used in bile salt formation and it's essential for brain development. And back in the 60s and 70s, before too much research was done on domestic cats, we actually had done studies, and you could see the negative ramifications pretty rapidly if the animals are not provided taurine. So why our obligate carnivores have to have taurine? So our cats, they can synthesize taurine, but only in small quantities. Once again, it makes sense because they evolved to be on the top end of the food chain. They're eating meat which has adequate amounts of taurine. So a lot of their taurine, they utilize that. It's combined with the bile salts, and the cats have incomplete reabsorption of taurine through the enterohepatic circulation. So you and I are able to reabsorb our bile salts with the taurine conjugated to it. Cats are not as efficient with that. So therefore, they end up excreting a lot of it with their bile salts. And when they are deficient, when they have inadequate amounts, we're going to have or form dilated cardiomyopathy. So this is basically heart failure, where the heart balloons at the bottom of the ventricles and the walls are very, very thin. And this was really common in cat food until it started being fortified with taurine in the 70s, once we discovered what the problem was. In addition to being needed for adequate heart function to prevent cardiomyopathy, it's also important for reproduction. So queens, which is a sexually mature female cat, has fewer kittens born and fewer survive to weaning if they are have inadequate taurine within their diet. In addition, you have the cats will get feline central retinal degeneration, so eventually they will become blind. So here's how taurine is synthesized. So it can start off with methionine, so our methionine can be converted to cysteine. So these dotted lines are the components that cats are not very good at or have limited synthesis. So the dotted line is limited synthesis. So our cats, although they have these two key enzymes, be able to tell me these two enzymes, cysteine deoxygenase and cysteine sulfonic acid decarboxylase. So be able to tell me these. They have them, but they're just not very active. So they have really low enzyme activity. So therefore that results in very little amount of taurine synthesis. So what do they do with all this added cysteine in their diet? They utilize it and break it down to pyruvate. And so that's providing energy for them. So that pyruvate's then able to go into the TCA cycle and give them even more ATP. So tell me these two enzymes that are involved, and why that they are required. Why taurine is required, and that's because they have these cysteine deoxygenase and cysteine sulfonic acid decarboxylase has low enzyme activity, resulting in cysteine being degraded to pyruvate instead of being converted to taurine. So here's a schematic of the dilated cardiomyopathy. So you could see the ballooning of the heart. So hearts are not supposed to be this round. So typically it would be more like that. So it balloons out, and you have these very thin walls that develop in the ventricular area. And once that occurs, it can't ever be fixed. So the only solution to the development of cardiomyopathy really is a heart transplant, which is not realistic for most pets. Now, humans, you're going to be able to get on the transplant list, but still not the best way to go about your life choices. So thankfully for humans, we're able to synthesize adequate amounts of taurine. Even though adult humans can make adequate amounts, infants are still not fully developed to where they can adequately make enough taurine for their diets or for their needs. Usually there's adequate amounts found in breast milk, and it was unfortunately discovered back in the early 80s that taurine had to be incorporated into baby formulas. So we have the infants, they ended up having low levels of circulating taurine, conjugated bile salts, and they also have very low levels of taurine excreted in their urine. So that gave an indication that they were deficient in taurine. So their body became very good at reabsorbing, and they became really good at reabsorbing taurine, so they didn't lose any. But we had many infants during that time that they still were subjected to taurine deficiency. So why is taurine important? So as I mentioned before with the cats, it's conjugated to bile salts, and it makes better emulsifiers. So usually our bile salts are conjugated to either taurine or glycine. Our taurine is the favored one because it makes better emulsions. Better emulsions means better absorption of the lipids. It's also a transmitter. So it's a neurotransmitter that is sending signals, so you'll have some neurological issues that develop if you have inadequate taurine in infants. It's also a photoreceptor or receptor in the retina. It's involved in glucose uptake, and it enhances the action of several hormones. So now we're going to switch gears a little bit and talk about proteins and their general structures. So we talked about amino acids, what's essential and non-essential, some of the conditionally essential ones. Now we're going to get back to overall proteins. What is proteins? So we have different structures of proteins. The first one is so our proteins are polymers of amino acids, and they are joined via peptide bonds. And then we have two different types of proteins. We have globular proteins, which are our enzymes all fall into the globular proteins. In addition, we have our fibrous, which is going to be our structural proteins in nails and hooves. And this is usually in the form of keratin. Our keratin is our main structural protein that we're dealing with. There are about 20 to 25 different keratin proteins that are utilized depending on the actual cell. So that is a fibrous one. Our enzymes are going to be globular proteins. So what does a peptide bond look like? Our peptide bond is what's formed between the amino group of one amino acid and the carboxyl group of another. So this is going off of the amide backbone of the amino acid. So here you have our oxygen off of the carboxyl end is combining with the two hydrogens. So we get the formation of a water. And then we have our peptide bond that's formed. So we get one molecule of water when we have this peptide bond formed. So remember, this is what we would refer to as a condensation reaction. So what is our overall categories of protein structure? So we have 4 different categories. Our first level is our primary structure, and this is what's making up our peptides. So this is our single chain of amino acids. Now typically once we have about 20 to 25 amino acids strung together, we start getting secondary structure. So our secondary structure is what forms when we get hydrogen bonds. So the hydrogen bonds between the R groups. And so that causes bends and curves. And we have 2 different types of secondary structure. We have our alpha helix. So this is going to be more in our globular proteins. And then we have our beta pleated sheet. So our alpha helix is going to be a spiral looking, and then our beta pleated sheet is going to be more like a S curve. Beta pleated sheets are going to be more prominent in our keratin proteins, our fibrous structural proteins. Our alpha helix is going to be more prominent in our globular proteins that are part of the cytoplasmic proteins and enzymes. Now, typically when you go into the tertiary structure, we're going to have a combination of alpha helix and beta pleated sheets. So our tertiary structure is where we get extensive coiling and folding. You have more ion interactions. These are typically stabilized by sulfhydryl groups, which are also referred to as disulfide bonds. So the sulfhydryl groups, if you can remember which 2 amino acids have those sulfurs in it, and those are going to be our methionine and our cysteine, which are really contributing to those disulfide bonds and that tertiary structure. Now, a lot of our proteins, once they hit the tertiary structure, they are done. They don't need any more. There are some other proteins that they are not functional, so we need to put multiple components of them together. So more than one tertiary structure protein together makes up our quaternary structure. So this is when we have multiple identical or different protein subunits come together to form one functional unit. A great example of this is going to be hemoglobin. And you also have antibodies. Are great examples of our quaternary structure. So here we have a picture of our different types of protein structure. You could see here this A is our primary structure, which this is what our peptides are. And as I mentioned earlier, once we get greater than 20 amino acids, we start getting secondary structure occur. So we're going to start forming an alpha helix, or we're going to get a beta pleated sheet. And then after that, we start forming the tertiary structure. So this is where we have a lot of the protein folding that's occurring, and that's occurring in the rough ER, inside the lumen of the rough ER. And then we're going to have our quaternary structure, where we have the different subunits. So here we have the example of hemoglobin, where we have the 4 subunits that are coming together to form the quaternary structure of hemoglobin. So our typical cells contain about 20 to 30% protein by volume, which that is a pretty significant amount. So this amount of crowding of protein actually ends up having a significant impact on the variety of protein related to the cellular processes, such as protein folding and stability. So a lot of times protein will lose the longer it's been working, especially enzymes. We're going to have to disassemble and remake that protein. So, and a lot of that has to do with the fact that there's so much protein found within the cells, and we have interactions with other proteins. So what is the protein folding? So protein folding, as I mentioned, this is what's making the tertiary structure. So it's what is providing the 3D structure of the protein. And this folding time really varies. So it can range from 1 microsecond to several minutes, all depending on the protein. As I mentioned earlier, this is all occurring in the lumen of the rough endoplasmic reticulum. And any protein that does not fold properly ends up getting degraded by a process referred to as endoplasmic reticulum associated degeneration. And your body actually utilizes this misfolding and breaks down this protein and actually uses it to get it back to the peptides, and your body puts those peptides on the outside of the cell, and that's how you determine self versus non-self too. So it does have some immune function too. So our misfolded proteins are not completely disregarded, that we do repurpose them. Now, if you have significant amounts of flawed protein folding, you can result in various diseases. One is amyloidosis, which incorporates Alzheimer's and Crutchfield-Jacobs disease. So you can, that's one of the problems with. So can we as scientists end up fix these misfolded proteins? Well, interestingly, Dr. Weiss from UC Irvine was actually able to unboil egg whites to where he was able to bring that egg albumin back to working order again. And it was always thought that once proteins are misfolded or quote unquote denatured, that they they can't be fixed. Well, Dr. Weiss and his group were actually able to reverse that. So, although it seems kind of silly, why would you want to unboil egg whites? Well, we can use the techniques that he used and start looking at those more closely. And can we better treat these people plagued with these diseases and reverse the effects of Alzheimer's? Can we cure cystic fibrosis? So that's a pretty exciting field and a lot of interesting things that is occurring right now, looking at the fixing misfolded proteins. And along with that, typically the when we're cooking food, we end up denaturing the protein, and this is some of the aspect of what Dr. Weiss is looking to reverse. And proteins are typically denatured when we are breaking down their tertiary or quaternary structure, and this occurs when they are subjected to heat. So when you are boiling your egg, it's pretty hot, so that's denaturing the protein. You're breaking those sulfide bonds and some of the hydrogen bonds. Acids, alcohols, heavy metals, so lead poisoning, mercury poisoning, those are heavy metals that are denaturing the protein. And then you have some other agents that also can denature protein. But it is interesting looking at, so it was always thought that this was, once protein was denatured, that it couldn't be fixed. But Dr. Weiss's research looks like we could potentially move to fixing some of these denaturations.